host range of plasmids, that when plasmids they are present in microbes, so whether they have narrow host range or wide host range. So plasmid usually encodes a few proteins that are necessary for its replication, and other proteins that are mainly the enzymes like DNA polymerases, helicases, or even ligases. They are provided by the host organisms. And those proteins that are encoded by plasmid for its own replication, those genes that are present very near to the ORI region or region of replication. ORI or region of replication, it is the point where DNA replication it mainly starts. So those genes that encode proteins for plasmid replication, they reside very close to the region of replication. So one can say that the essential genes for DNA replication, they are present very near to the region of replication. And other part of the plasmid, it is not required for plasmid replication. So we can delete those part of the plasmid that are not required for DNA replication and can replace it with foreign DNA or gene of interest. So now we can say that the host range of plasmid, it is mainly determined by its region of replication. So I can quote one example of the plasmid like COL-E1. It is having restricted host range and it can transferred within a few enteric bacteria like gram-negative E. coli or the Salmonella or Shigella. Whereas other plasmid like RP4 and RSF1010, they are having slightly broad host range. For example, RP4, it can be transferred in variety of gram-negative bacteria. So DNA can be transferred from one gram-negative bacteria to another gram-negative bacteria that are more or less taxonomically related. जबकि जो दूसरी एग्जांपल है RSF1010 की, तो ये जो प्लाज्मिड है इसकी होस्ट रेंज कंपैरेटिवली ज़्यादा है क्योंकि ये ग्राम नेगेटिव से ग्राम पॉजिटिव में भी शिफ्ट हो सकता है. So the transfer of DNA, it is from within gram positive or gram negative. So other plasmids that are isolated from gram positive bacteria like the Staphylococcus aureus, they are also having broad host range because they can transfer between variety of gram positive bacteria. Overall, we can say that the host range of the plasmid, it is determined by its region of replication, and if plasmid it is having more genes or that can encode variety of proteins that mediate its replication, then the host range of the plasmid, it is comparatively broad.